Statistics you guys have heard a lot about. And so I'm going to focus less on the methods and stats and a little bit more on kind of the practical implications of those statistical choices. And so, again, these are all detailed in your protocol so that you don't get to the end of the study and go, what did we want to look at? What was it that we were going to analyze? We had this great idea, and then you go back to your application and you're like, yeah, we described that in 2 paragraphs because that was all the space we had. So this is where you have the opportunity, lay it all out. Your analytic plan can be done so that as soon as the data is clean, your biostatistician can run it because you already know what you're going to do. So it saves you a lot of time in the end if you have this all spelled out ahead of time. And more importantly, what we're seeing in the open science trends and for rigor and reproducibility is that people are having to put their a priori hypotheses and in some cases analytic plans out there and before they even start enrolling participants. And so journals are now comparing back to, well, what did you say your primary outcome was going to be? And is that actually what you're reporting in your primary paper here? So it's important to get those things specified and detailed. I mentioned before that when I think I was talking about blood pressure and how you might collect that. So this is an area of your protocol where you define really clear how you're defining your outcomes. So for cancer studies, you might have an appointed panel of experts that actually review the pathology slides to determine whether or not a person actually has cancer or not, or meets the definition of non-alcoholic steatohepatitis for a liver study. You may actually have a group of individuals who review those blinded and make a determination as to whether they think that disease is present or not in the sample that they receive. They might see an imaging scan and make a determination as to whether or not, what is the size of the tumor? Has it shrunk or not? And so they actually give the measurements and those kinds of things. So you want to define how, for your study, you're going to define your outcome. And I really do encourage people to use the standard definitions in the field, the agreed upon either diagnostic criteria or methods that have been published by a given field. And so one way to find those is to look for other large clinical trials that have been done in a particular area and see what definitions they have used. The clinicaltrials.gov resource that lists all of the trials that are ongoing is a great resource to be able to look at inclusion and exclusion criteria for other studies so that you can see that you're enrolling the same kinds of patients as other trials are, and also the definitions of their outcomes so that you can use similar outcome definitions in your trial, and then you're not creating something de novo and totally new that you've come up with that would be difficult to include in a meta-analysis later because you didn't use an agreed upon definition that the rest of the field is using. So, for example, the studies that I have listed here are the Women's Health Initiative, the Systolic Hypertension in Elderly Program, which has definitions for how do you define hypertension in older adults, the studies of left ventricular dysfunction, the SOLVED study. They have very clear definitions of how they've identified what is an MI, what is a myocardial infarction, what is a heart attack. And so those are really good resources to look at those very large trials that have spent a lot of time determining how they're going to make those determinations as to what the definitions are of the primary outcomes. One of the things that can be a challenge is specifying not only what data you're going to collect, but in what units you're going to collect it. I used to show a slide, and I don't think it's in this slide deck anymore, where people had collected height and weight and different measures, and some of them were in, weight was in kilograms, and sometimes it was in pounds. That you had different staff writing it down in different ways, and if you tried to merge that data, you get really skewed information and not really know how to interpret the summary data. So case report forms are what we typically collect the data on, either electronically in electronic case report forms or in paper copies, which we're seeing less of now. It's really important that they include the units as to how they're supposed to be measured. And if you're collecting it electronically, you can actually create limits in your system so that won't allow you to put in an adult's weight at 10 pounds, that it just wouldn't allow you, that you would have ranges as to what's acceptable. So those are elements that are very important. How you're measuring it. We do occasionally get investigators that want to rely on self-report for height and weight, which, if that's not a primary outcome and it's just a kind of a demographics information that you're collecting, it's fine. But if you're planning to use BMI and calculate BMI from that information and use it as a confounding variable and to adjust for it in your analysis, you probably need to get accurate and measure height and weight. And certainly if you're ever going to share that data for secondary data analysis, it would be important to label it as to whether it's measured height and weight or whether it's self-reported height and weight, so that the data couldn't be inappropriately used thinking that it was actually measured as opposed to self-report. And the measurement piece of this is this reproducibility theme comes up again. Yeah, is there a question? Okay. Making sure that you are collecting the measure in the way other people do it as well. So I gave you the example of blood pressure. So for most studies, it's done sitting. It's done after a period of rest when the person is not talking, is not eating. They get quite frustrated because they don't like to sit for typically about 5 minutes. And people's attention span, that is forever for people at this time. But that's the typical. If you need to do something for your study in a different way, if you actually want to get a reading on blood pressure when someone's exercising, it's really important that you label your variables that way and make it very clear as to, well, how much exercise do they need to do, and at what heart rate are you trying to get them to, and what blood pressure would you try to get for that. So detailing all of these aspects in the protocol is really important, so your staff can collect the data the way that you want it collected. So you've likely heard some issues around study instrument surveys and how to think about using them, the importance of making sure that they're actually reliable and valid the way that you're using them, and that they're actually sensitive to change. So there are some measures, particularly in the psychology literature, which we talk about as state or trait. So state is the current state that you're in, and you would expect that to fluctuate from day to day, time to time. How anxious are you giving a talk versus how anxious are you relaxing and getting a massage? Likely very different depending on who you are. One may make you much more anxious than the other depending on who you are. Whereas trait variables are thought to be quite static and not change. If you choose an outcome measure and you're not very familiar with the different instruments and you happen to choose something that is a trait variable that's not going to change, then it's not a very good outcome measure to see if your intervention is going to have an influence on that. If you really do think that trait variables and trait characteristics of individuals are something that are static. There certainly is a literature that's pushing to think that there are some interventions that can shift people's trait tendencies in terms of their characteristics, but for the most part, people think about state as something that you would want to potentially measure as an outcome variable because it is variable, versus a trait that is something that is just a characteristic about the person. So those things are really important to think about as you're selecting your outcome measures. Over what time period did things change? So some instruments are really only developed to be used every 6 months or once a year. Some of the stress characteristics, the perceived stress scale is one. Some of the items on there are, have you moved in the last year? Well, that's not going to change very likely in 6 weeks. So that item is going to be pretty static for most people on that particular scale. Have you lost your job? Have you had a death in the family? Those things are not something that on a day-to-day basis shift very much for a given individual. And so a more daily stress measure might be something better if you're looking for changes in whether someone's stress levels change over time. The other thing that we're seeing more and more of with outcome measures, most of the particular survey instruments were developed as pen and paper instruments. And we see them more and more being shifted to being collected online or being collected through EMA, ecological momentary assessment. So they ping you with a text and ask you a question or two in real time, wherever you are, to find out currently where you're at, how do you feel. And if the instrument wasn't validated to be used that way and to be assessed that way, then it really needs to be validated to be used that way, because there could be confusion around the language, there could be a lot of problems with how reliable it is and whether you get the same response every time you ask the question. And so it's not that it can't be done, it's that someone, before you use it as your primary outcome measure, make sure that you're choosing an instrument that has been validated and is found to be reliable in the way that you're actually using it. And this comes down to kind of, is it a good measure for you, for your study? As this says at the end here, there may not be published data on it, but that might be an opportunity for you to work with a psychometrician to do the validity study on delivering it in a different way. We need more valid and reliable measures through a variety of different delivery methods, because patients and participants in research studies get very frustrated filling out hours and hours of forms. And you've likely heard some about the computer adaptive testing that is available through some of these instruments now, where an individual takes an online, does a set of questions, and it calibrates to the individual the way the GRE is done this way. So it hones you into your score based on what your responses are, rather than asking everyone the exact same set of questions. It's really customized to the individual who's responding to the questions. It can dramatically shorten the number of items that an individual has to answer to respond that way. But it's very foreign to an investigator, and I think they're getting more comfortable to this, but to know that not everyone answers the same questions is something that's very different than a typical standard set of a 27-item questionnaire, for example. So, as I mentioned, these are the elements that you certainly want to include in your protocol in terms of the general design issues, your treatment assignment. The randomization procedures is something that's really important, and we're starting to see, particularly at my center, more and more interventions that are delivered as group interventions. And it's very important to specify whether you are individually taking an individual that decides to participate, and there's a random chance as to them participating in Class A versus Class B, or whether you've created a random allocation of classes, and depending on when that participant happens to call, they get the next class that's available. And as you can imagine, if that randomization allocation sequence becomes known to study staff or to the people enrolling participants, it may influence who they decide to participate. So if treatment A is a surgical procedure and treatment B is a watch and wait, they may hold off. I mean, this is an extreme example. This is not likely to actually happen in a trial, but there certainly is published literature that if the staff is aware, they may even unconsciously not refer people to a study if they know that the next treatment allocation is for a watch and wait. If they think that patient really needs treatment, they may actually not even refer patients. Whereas if they know the next treatment allocation is to the actual intervention, they're likely to refer a lot more patients, and potentially much more severe patients that they think would respond or do well with that particular treatment. So the blinding of the delivery and the sequence of class delivery in particular is something that's really important if you're doing a random class allocation, which we're starting to see more and more. And the same is true with other types of cluster designs and that need to be taken into account. The other issue that comes up that I don't think gets enough attention is the fact that when you randomize the participants in a study is really important in terms of your study flow. One of the things I'm going to talk about, and I'm sure one of the terms you've already heard, is intention to treat analysis, which is everyone randomized gets analyzed. And because of that, you want to shorten the time and the wait period between when the randomization is done and when the participant actually starts the intervention. So I've mentioned a few times that we often have these class-delivered interventions. Mindfulness-based stress reduction is one of them, but this could be true for a group-based cognitive behavioral therapy or other group-based visits for diabetes management. And if you have to wait until you get 15 or 20 participants before you can start the classes, if the first time someone calls you and you determine that they're eligible, you decide to randomize them and you say, okay, you've been randomized to this class and it happens on Tuesday night and our first class will be on April 25th. And then you wait and you contact people, you're going to lose a lot of people. And so you run the risk of having a lot of people drop out of your study after they've already been randomized and you now have to include them in your analysis. If instead you screen people and you let them know, okay, our classes will be starting around the middle or the end of April, we'll call you again at the middle of the end of April to make sure you still want to participate and confirm some of the characteristics that nothing has changed and you're still eligible for the study. The study is planned, your first class is April 25th, and you let them know the class will either be on this night or on this night. Please make sure your schedule is clear so that you can participate in the class. You call them ideally the day before, if you can. You wait really as long as possible to be able to confirm that they're still eligible and then tell them which class they're going to go to after you confirm they can still attend the class. Because so many things come up. Oh, my parent, I have a sick child that I've got to do all of these visits with, or oh no, my neighbor fell and broke their hip and now I have to help with care for them, or my grandchild needs aftercare and my daughter got a new job and so I have to pick her up instead. So many things happen in life between the time someone decides to participate and potentially when a class might start for the class. And so putting off that randomization until the last possible moment that's feasible for the study to pull off can save you tremendously in missing data in the long run for your analysis. So that's one of the things that I certainly wanted to mention tonight. The other thing that I'll mention is that if you have interim analyses, you include those in these studies in your protocol as well. And hopefully you've had some information about, really only doing interim analysis and having stopping rules if it's absolutely necessary for your study, if there's a real reason to want to assess for futility and end the study early. And certainly your full analytic plan needs to be included as well. I think you guys have heard a lot about sample sizes and the importance of basing your sample size on your primary outcome and the primary hypothesis that you're going to test, and that you've got enough information in which to base that. And there's many different ways to analyze data. My take-home message as an investigator and someone giving advice to investigators is have a really good biostatistician that you work with from the beginning all the way through your study. And don't just hand them your data at the end, because it's likely they won't be able to do the analysis that you hoped they would be able to do. You want to involve them from the very beginning to make sure you're designing your study to actually answer your hypotheses and you're collecting the data that they need to look at the outcomes and control for any potential confounders that may be measured in your study. Again, I'm not going to go into a lot of the details, although I will emphasize the point on the analytic plan that you do need to have a very clear process laid out as to how you're going to handle missing data, what you're going to do about it, how your statistician, are they going to try to impute based on other data? What is the method that they're going to deal with the issue of missing data? And how are they going to evaluate whether it's missing at random or whether it's actually informative missingness and that you had differential dropout. So your waitlist control that you thought would be great well, nobody came back for their follow-up visits, and so you have very little follow-up data on your waitlist comparison group. It can be a definite problem in studies that have very different types of interventions for participants and different engagement of the participants. I mentioned the interim analysis, and the biggest thing here is that you want to be able to analyze your data so that it will have an impact. And so all of these elements of including these pieces in the protocol ultimately get you to the place where you have good quality data that's been collected consistently across all participants, that you've included only participants who are eligible for your study, and that if everyone followed the protocol all the way through, you have very little to no missing data. So you have a great quality data set on which to base your analytics and then conclusions of your study. The more missing data you have, the more difficult it is to interpret the results of your study and make really conclusive statements about whether or not your intervention was beneficial or not. This I mentioned briefly just because it's this concept of if they're randomized, it is someone that you need to include in your intent to treat analysis. I'll refer you to your biostatisticians about whether or not it's wise to do modified intent to treat analysis. There's a lot of differing opinion about that. Certainly we do see per protocol analyses to see, and oftentimes these are explored as different types of sensitivity analysis to see whether there's a huge impact of whether or not people are compliant or adherent to the protocol in terms of their benefit with the treatment itself.